Okay, let's get started. Good. This was homework for today. Another bit of playing with smoothing, this time really the real biological data, and in the end we want to get to p-spline. Now, two of you have sent me some email with some code. How about the others? Who has given it a try, and how far have you got? How far have you got? Okay, let's see. Pretty. Okay, then let's see how far we get. Let's simply start from the beginning. Yeah, I haven't done it yet myself, but maybe that's quite good because then we can develop it together. So I start in RStudio. So the first thing is, some of the two who sent me an email, you both sent me R code, but without images and so on. One commented that it didn't work. So let's see how this works. And you see the first thing, when I start a new file here, I don't start an R script, but I start a quarter document. I usually just click on create empty document, and then I get here this empty document, and we have a switch between source and visual. I don't like the visual because it's new and still a bit buggy, so I stay here with the, you see, with the source mode. The source mode means, you'll see the difference is that there's a couple of things which are explicitly written here if you use the source mode, and if you switch to visual, it changes a bit. We need a title, so title should be probably homework. Homework 5 was that. And now here in this notebook mode, I can insert these code chunks, and everything I write here in the code chunk will appear below as a result, and everything I write around here will appear as normal text. In the end, I can click on render, and I have work 5 solution. And I get an HTML file which contains what I've typed so far. And in that way, we can now try to get our homework together. So let's see, what was the first step was that we were supposed to download the data set. I, of course, already have it downloaded. And when we want to load the data, as shown here, so we simply use the same code. So here maybe Ctrl-Shift-E. Just put this code in here. I use the read empty-x function, which I think was from so let's use Sera. I put that in here. And because when I load Sera, you see it prints all this stuff here, and I don't want to have it in my final solution. So I say here, message equals false. So the way how these chunks work in Quarto, you always have code chunks which start with a triple backtick and end with a triple backtick. And then you have here in curly braces the language, in our case R, and that means what we type here will be ran through the R interpreter. And we can give so-called chunk options which tell us how this chunk should be displayed. And I can put things, for example, here like message is false, don't show all the messages. Or here you see all the other kind of options I can give. And of course, there's something about it. There's also eval for evaluation, whether it should be run at all, whether results should be printed. So I now use this here. And now you see here I get my library. Now I can load here my data. And in the end, I get my count matrix. Let's give it a moment. Okay, now it's loaded. Often it's a good idea to then here quickly have a look at this thing. Seems to be a nice matrix. Let's look at the dimension of the thing. Count matrix. You see it's 20,000 genes, 18,000 cells. That looks reasonable. Now, next, we wanted to do the standard Seurat pipeline, it says here. Perform the standard Seurat pipeline simply by copying over what we have here. So let's do this. Data. Run standard Seurat pipeline. And I put this in here. And again, I say message is false because I don't want to get all this stuff which it produces here. And could not find function. That must be tidyverse that I need for that. Library tidyverse. So. And now it runs through all these things. And you see, it does all the stuff that you've always done, find the neighbors, find the cluster, run the UMAP. And the first thing is we want to inspect the cluster. So while it's running, I already type this. Look at clustering. So, UMAP plot. Sorry. Oh. Here is my UMAP plot. What I want to do is I want to be able to quickly see which color is which. No, it wasn't labels. What was it? Anybody still remembers? Otherwise, I quickly check here what I've done. Label is true. Okay. And the UMAP plot function of Seurat has this annoying habit that it makes the axis with non-unit aspect ratio, so that it always looks a bit squeezed, and I don't like this, so I tell it to equalize the axes, and now I get this thing. And now we can here look at our lineage, and as we've done last time, we simply define all the parts which are here part of the lineage, 10, 0, 13, 9, maybe 14, maybe not, I'm not quite sure. What have we done last time? Have we taken 14? Yes. So here is all the clusters. Let's take them and use this as the lineage. And I just quickly want to check whether I got the right clusters, not that something changed. So what I will now do is to check that is I first need the UMAP coordinates to redo this plot manually. And that is, I think it's called embeddings in Seurat. UMAP. Yes, these are our UMAP things. I change this into a tibble. I add to this the clusters by saying, I don't know, have you guys worked with tidyverse before? So tidyverse is a useful little thing. Maybe it is for those who work with Python might recognize that it's pretty similar to how you work with pandas. The idea is it's a package which has functionality to manipulate tables. And whenever you have data tables, you can easily do this manipulation. It has a bit its own nomenclature. For example, a table is called a tibble. And this here is, as you can see here now, a tibble with two columns. I can also tell it that it should keep these row names and maybe call them cell. And now with this, you see I now have a table with three columns, one for the cells, one for the UMAP, and the other for the UMAP. So I took this embedding stuff out of Sera, which is the UMAP coordinates, and then I turned it into a tibble so that I can use the tidyverse functionality. And tidyverse has a couple of, let me quickly show you how this looks like on, let's say, dplyr sheet. Here you can see all these different things that you can do with tidyverse, and it's quite convenient if you quickly want to change tables. So you see here the logic, you have columns and rows, you have this pipe symbol, which allows you to write a function call like this, such that the first argument of a function is not put in the function call, but before with this %. So this is an alternative way of writing this. And on the first hand, it looks strange why this is useful, but you will see in a moment why this is useful. And here are now different kinds of functions. You can group your data set in different groups of rows according to the value of one column. And then you can calculate summarization statistics, like what is the mean of the another column per group or something like that. And of course, this is something that you often do. Those of you who've ever worked with SQL will recognize that it's very simple to the logic that you know from relational databases. And here now, what I now use is a function which is called mutate, and it simply adds a column. And I call this column cluster. And now I have to somehow find in Sera, in my Sera object, the cluster identity, and I think it's here. Now you see I have mutate always adds or changes a column, and now I have added a column which took here the Sera cluster out of it. The next thing I can now do is again mutate and do something like in lineage. And for this I ask is the value here that's here in cluster within all the values that I've put here. And now I get here trues and false depending on whether this cluster is in this list of lineage things or not. And here you see why this % operator, this pipe operator is useful. It essentially always takes the data, the table, and pushes it one command to the next command. Syntactically, it takes, it uses whatever you pipe in here from the left and inserts it as its first argument here in front of the actual arguments, because all these functions here, they expect a data table to come in. And last, I send it to the ggplot function, which you might know as the standard plotting function. Or R has different plotting systems. There's the base R plotting system, which has always been in R, and there's the ggplot plotting system that's part of tidyverse. And ggplot works the way that you push a table into ggplot, and then you can choose various plotting things, for example, geom point for a scatter plot, and each of these plotting things has so-called aesthetics. In our case, geom point has aesthetics X, Y, and color for X, Y, and color, and we have to tell now a ggplot from which column from which columns it should take these aesthetics. And it should take the X coordinate from the column labeled UMAP1, with Y coordinate here, and for color, it should take it from in lineage. What's the point of this now? So if you haven't used tidyverse on ggplot before, you see here the general framework of this plotting scheme with ggplot, and it's quite powerful, because you can do a lot of different kinds of plots. But the easiest way is you somehow construct a table like this, and when you push this table into the ggplot function, and when you add to the ggplot function all the components of your plot. The moment our plot just has one component, namely a layer of points, and each of these components has a word value which starts with geom, and then comes what it is, in that kind of points, and it has this aesthetics block where you have to assign column tables to the different aesthetics. Aesthetics is a stupid word. It here simply means the different degrees of freedom of a plot. If I do this now, I get here my plot. You see the points. It used X and Y, made points at each point, made some of them a bit thick, some of them, the lines are all a bit, the points are all a bit big, so I make them smaller by saying size is 0.1, and you see this aesthetic is now outside S because it's not a table column. Now it looks like this, and then I want to add another speciality to my plot, namely again, that it should make my X and Y axis here, the length here and the length here, the aspect ratio the same. With this now, I have now clearly shown which cells I consider part of the lineage and which not, namely all the blues are part of the lineage and all the red ones are not. This was just to double check that here with the lineage cluster, I got the right kind of clusters. So let's write this here. Select clusters that form the lineage. Plot this to check selection. So now I've done this, and the next thing we can now do is we find this principal curve through the lineage. And again, I think before we try to do too many complicated stuff here, we can also here just jump to the lecture principal curve. Okay, where was the principal curve? Here is fitting the principal curve. Okay, let's see here. I just copy that thing over to the other stuff. Ctrl-Shift-I, put that in here. Now we see what's happening here. It takes here, it takes here, by the way, what I should show, whenever I copy such a code, I want to know what the code does. And a good trick to do that is the following. In RStudio, you can mark with your mouse parts of a command, like this here, and press Ctrl-Enter, and then it executes only this part. So if I have this complicated command and I don't quite know which data it is used, and I want to know what does actually this part works, I press Ctrl-Enter, and then I see, embedding soy, PCA produces a table with these things. And then comes here this question, soy $Sera cluster. What is this? This is a vector which assigns to each cell a number of which we can check if it's in here or not. Yeah, I remember, notice here we have a lineage cluster 2. Ours is called lineage clusters. But if you remember from the lecture, back when we decided to exclude these two, 6 and 11, because they are sort of here in the cell cycle, and they might confuse the whole thing. Let's remove 6 and 11 for the moment, so that we have the same as before, and afterwards we can try to remember and put them maybe back in. Now with this, you see what I've now removed is this extra thing, so that there's only one way to get from here to here, which avoids us the principal curve to get wrapped up in this thing. And now here we can check what our principal curve does. We have here the PCA coordinates. And as you can see here, if I subset with this, I get only those rows of this big matrix, which belongs to cells in the lineage. And when the matrix gets a few less rows, and I give this to the principal curve function and tell it I want a principal curve with 10 degrees of freedom. And what this means is we'll discuss next lecture, but for now already in brief, what this principal curve function does is it tries to find a spline for each of the coordinates. For each of our 20 principal coordinates, it tries to find a spline which describes this as a function of a position along the curve. Here it says trace equals true, so that it tells us what it does. And the approx point is also something we need so that it doesn't take ages, namely that it picks, instead of doing it on all the 13,000 cells, it picks 1,000 cells at random and only runs the curves through these. Because if we pick them at random from all of them, they are probably a reasonable representative sample from the whole thing, and the curve will look the same as without that. I run this, and now it takes a while. Now I've removed and we put that trace equals true back in so that we get an output. And you see here it makes its curve, and it shows us always how far the data points are from the closest curve point and adds always up. So this is our mean squared deviation, our sum of squared deviations of the data points from the curve, and it tries to bring the curve in the middle of the data point so that this gets smaller and smaller. And when it doesn't get much smaller, it declares convergence and stops. Actually, I think it always only does 10 degrees or so. So now is a good time to investigate what we got back from this thing. You see, I collected this thing in this PRC object, and let's have a look at it. If I press it so, it tells me the component of this PRC object, and we see it has a slot called $S. Goes on and on and on. A slot $ord, $lambda, and what's the last one? Anyway, this gets a bit annoying scrolling, so there is a useful function in R which is called str for structure. And if you give it a list of many things, it just shows you the starting point of each of them. So here it gives us, one says PRC is a list of 7 elements called S, ord, lambda, dist, and so on. And here we can read what we get. We get here first S. These are the curve points. It tells us for each cell what is the closest curve point for this cell in this 20-dimensional thing. This is why we now get here this 14,000 elements and this matrix of 14,000 rows for the 14,000 cells in the lineage and these 20 coordinates. The next thing that we have is here the order, which tells us in which order the cells appear on the lineage. So the first cell in the list is the 6,400 cell in the lineage, and so on. Lambda tells us the distance of each cell from the starting point. This is the total distance of all of them. Converged tells us whether it converged, and it isn't happy with the conversion and says it could have gone on a bit more. But it seems that we stopped it after 10 iterations. Let's quickly check why this is. You can see here that this thing has a max iteration parameter which is set to 10. So maybe, as we've seen, it only takes a little bit. Maybe we should tell it max it is 30 or so and check a bit longer to see whether this helps us to get a better fit. Or maybe this whole thing is already good enough. So did everybody get that far? Because so far we've just used the code from the lecture. So any problems that occurred to you trying up to here? Good, because now it sort of gets interesting. Now we can try to do actual work. And before I look into the actual parts of this lecture, in the actual parts of our homework, I want to play a bit with the PRC object, so we learn how to understand such an object. So let's see. Oh, it stopped here after 13 iteration. It seems to have decided that now it has converged. You see now it says converged is true. So the first thing I want to do is to visualize the curve. And for this, what I will do is I take our UMAP plot from up here. And this time I want to color the cells according to their position in the curve, so according to their lambda value. How do I do that? First, I make, again, here I have my UMAP coordinates because I want to display this in the UMAP, and now I have to add a third column which tells me this lambda here. And to do that, I do the following trick. I can make here this S $lambda, sorry, principal curve $lambda. And if we look at this, you see this thing is now simply a vector which assigns a number to each cell. And we want to add this thing to this table. But the two things don't have their cells in the same order. Why is this? Because remember this table here, if you look at it, this table has 18,000 cells, while this thing here has only 14,000 cells. Why that? Because we have excluded all the red cells here. So if I now want to take this vector and add it as an extra column to my table, I have to be careful that I don't accidentally max up the cells. And with tidyverse, this is easy to do. The first thing that I do for this is that I run this thing here through a function called nFrame. nFrame takes a vector and turns it into a data frame. Data frame is R's parlance, is what R calls a table. And here you see now we get our data frame. And instead of a vector where each element has a name, it's now a table with two columns. And I can also give this thing here two words to tell it how it should, what it should use as headings of these two tables. And to do this, for the second part, of course, I call it lambda, because that's where we got it from. And for the first one, I call it cell. Why that? Because now I have this data frame here, which you see has 14,000 rows, and each row is identified by this cell ID. And now let's look at this thing. This is also a table, but with 18,000 cells, and it also has a column cell. And by this, we can now join these two tables by telling it that it should make a new table, which is a combination out of these two tables, such that only rows which agree in their value in the cell column sit together. And I do this as follows. I say left join. And left join wants two arguments, namely a table and another table. And they are called the left and the right table. And it wants an information about how to combine them. Namely, I want to combine them by with respect to the cell column. And if I now do that, what you can now see is I now get here a new table with 4 columns, which is UMAP 1, UMAP 2, and lambda. And because the second table was missing some of the cells in the first table, some of these values are here marked as not available. So that's a nice thing we can do with this left join. And you can already see there's an asymmetry here. It's called left join. There should also be a right join. And it's called left join because we start with a left table, so with a font past first, and this is a fixed table, and to this we add rows from the right table. So any cell which is missing in the right table will be put as NA. But if there had been a cell that appears only in the right table, but not in the left table, it would have been dropped. So left join means make a table which contains all the rows of the left table plus the appropriate columns from the appropriate columns from the right table. You can do it the other way. You can have an inner join where it uses only those which appear in both, and a full join where it uses everything which appears in at least one of the two tables. If you've ever done anything with SQL or with relational databases, you might be well familiar with this concept of table joining. Who has worked with table joins before? All the computer scientists, hopefully, certainly. The mathematicians, well, now you know this general idea. And you see, this is the reason why tidyverse is so popular, because these kinds of things get really ugly if you want to do it with other ways, and you easily get confused. With all this now, I can now take my plot that I have here and can move it again to my ggplot, and again I gave it an aesthetic and say x should be umap 1, y should be umap 2, and the color should this time be taken from the column lambda. And again I say I don't want two big classes, and also I say that I want this here. And if I do that now, as you can see, now each cell is, we get the same plot as before, but now each cell is labeled according to lambda. And the cells which we had excluded stay gray because they don't have a lambda. So now we can see what this, our principal curve has done. It has brought the cells into an order such that the cells start here, go round, 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 and you see the lambda value increases from here over here to here. So that is our first thing. We see now for each cell, where it is put in the principal curve. Remember, this is we are working in this 20 or 30 dimensional PCA space. And in this space, this is a curve which goes through, and each cell is projected onto this curve. And the color that we here see is the distance of the projection point from the start of the curve measured as a curve integral along the curve. And as you can see, we get it out here. We can also check for each cell how far it is from its corresponding data point. How would we do this? Anybody has an idea? I want to know, because obviously if I have points like this, and I make my curves through it, when these points are well described by the curve, but the points out there are not so well described by the curve. How can I indicate on my heat map, on my UMAP, which points are well described and which aren't? So I want to indicate, at the moment the color indicates for any cell the distance from the starting point. At the moment, this here is lambda, this distance. What I now want is this here. And what I have is, of course, the cell's original components in the PCA space and the cell's projection point. Where do I have this? The cell's original components I have in my embedding thing. Embedding str, PCA. These are the cell's original coordinates in the 50-dimensional space. And PSC$S, these are the cell's projected coordinates. So how do I now, yeah? Okay. Let's try to do this. First attempt. Let's make here a new thing. First attempt, we take here the $S. So these are the projection points, and the other thing was embeddings soy, PCA. These are the original point positions. And now we subtract these two matrices, and it says error, non-conforming arrays. Why that? What went wrong? Whenever you get such an error, a good idea is to first have a look at the two objects which we have and inspect them again. So what I now do is I use this str function on embeddings and on $S just to see what they are. And we see for the first one, it tells us, so this here is the first matrix, the matrix with PC coordinates and with the cell's original coordinates, and these are the projection points. Let's, just so that we understand this output of STR, let's have a look. It tells us that it's a numeric data object with dimensions 18 thousandths for the rows and 1 to 20 for the columns, and here a couple of numbers. And furthermore, the rows and columns also have not only index values, but they also have names associated, namely the 18,000 rows are labeled with these 18,000 strings, and the 20 columns are labeled with these 20 strings here. And then comes here the output of the STR on the other thing. So what's the crucial difference between these two? Why can't we subtract these two matrices from each other? Yeah. Exactly. So what we have to do is that we first have to reduce this thing to only the 14,000 columns which are in here. How can we do this in a convenient way? That's now something which maybe if you work with other language you're not used to, but this is where these labels come in that I just mentioned. You see here, our matrix not only has numbers, it has also labels for the rows and columns. And if I say row names of this here, I get these labels. These are the labels of the 14,000 cells that we do have. And I can use them to index the other thing by doing this here. So what does this mean? Square brackets are indexing in R. So it means from this matrix, take these rows, the rows with these labels. So here we have the names of the 14,000 cells we want, and we use them to select the corresponding rows from this one. And if we do this now, we get the 14,000 row matrix. Does this make sense? So that's something which is quite convenient about R, that with these labels that the rows and columns have, you can do something like that and tell it from this matrix, pick only the rows with the following labels, namely the labels which appear in the other matrix. And now I can subtract the other matrix. And here this is now the and what is this matrix now? How would you describe this matrix that we have here? What have we seen now? Yep, thanks, but nobody else has any answers? Who's still with us? See if everybody, we subtracted two matrices. What is the first matrix? Who still remembers what the first matrix was? Yeah. Yeah, and what's the second matrix? Okay, so the first matrix are the original coordinates, the second are the projection on the curve. So now that we have subtracted the two matrices, what do we have now in our matrix? No, not yet the distance. The difference vectors. The vector from here to here, all these little vectors. And what we now want to get is the length of these vectors. Well, how do we do that? Mm-hmm. Okay, I take this thing, put them in, and square the entries. Now they're all squared. Now I take the row sums of this, like this, and now we have for each matrix its squared distance. And of this, we take the square roots. So, and what do we do with this now? I wanted to remember this, I wanted to make here this plot, and I wanted to replace this plot with a plot where where instead of the position along the curve, we see the distance to the curve. So what do? I take the code that we just had, copy it over here. And before we had this thing here, and I replace this here with this here. Try and see. And maybe I should call this now, instead of lambda, I should call it distance. Maybe to make it a little bit less opaque, perhaps I should break this into two things by, oh no, let's keep it like this. And here, now it should be colored by this distance that we've just put in here. And now we see here the distance. We see how far are they all from this, but it looks all the same. Anybody has an idea how we can make this plot a bit more useful? Mm-hmm. Exactly. Let's make a logarithmic color scale. Plus scale color log. Oh, doesn't have anything. I think it works like this. Okay, now we have here a logarithmic color scale, and we can see everything here in the middle. This black part works quite well. This blue and this here is already a bit away. This is also a bit away. Now we have a good feeling of where the curve goes, which points are really close to the curves and where the points are a bit further. It seems here everything is rather tight, and here it becomes rather spread out. Here. So let's see again what we've done. We've used here the UMAP coordinates, which we took out of the Seurat object. Here we calculated the distance. When we put these two tables together, using the cell labels to get things right, and then we plot it, and we used here the scale color continuous to get that part. But the one thing I might change here is gradient n, comma, colors is RJE. So now I've changed the color scale to a color scale which is called the cube helix color scale, because that makes it much easier to see where things change here. You can see using a good color scale can become quite important, because before you haven't seen much, and now everything is I think, much clearer. This distance here we couldn't see before. What's the cube helix color scale? That's a rather neat idea, which comes from an astrophysicist and a guy called David Green from Cambridge, who, as you can see here, was a radio astronomer and wanted to depict maps of radio intensity. And he came up with this idea. This is his color scale, and here you see how it constructed. This thing is the so-called color cube. It's the three-dimensional unit cube where we use the three axes as indicating red, green, and blue. You know that on a computer, every color is described by giving the intensity of the 3 pixels on your screen, how intense, how strong the illumination of the red, the green, and the blue pixels should be on a scale from 0 to 1. And as you see, what David Green's idea was to make a helical winding around the spatial diagonal of his cube, because this corner is black, all pixels off. The opposite corner is white, all pixels on. If you connect them directly with a line, you go through all the gray values, because the diagonal will always have all 3 colors of the same intensity, and then it looks gray. But if you don't stay on the diagonal, but go a bit away from the diagonal and spiral around it, then not only the brightness of the color will change, but also the intensity along also the hue, the color, the shade of the color. So when we go once around the spectrum, you know, here the color wheel, which is the rainbow spectrum plus the purple colors, and get back here. And that's what you see here. This is the formula he came up with, which is essentially 3 signs, which are shifted by a phase angle of 2/3 pi, and overlaid with a linearly amplifying thing, and then here also overlaid with some envelope, which makes sure that we start at 0 and end at 1. That's a pretty neat idea that he had here. You can also tell the cube helix function that you want more windings or less windings, depending on what you want. So, as you can see, even things like choosing color contains geometry. Because here we have now used our understanding of standard 3-dimensional geometry to describe this helix in a cube in order to obtain a useful color scale, which allows us to easily distinguish different kinds of different values in this heat map here. So, but that was a bit of a detour now, because what we actually wanted to do is, let's look at the homework. What was the actual job now? Now create a data frame with 1 row per cell and the following columns. Okay, let's do this. Create data frame. Data frame, as I said, is just our language for a table, and in tidyverse we use the tibble function to make them. So first we want the row names of a server object, row names of SU. That is, as you can see here, the gene. No, cell barcodes. I wanted the cells. And these are, of course, not the row names, these are the col names. I have written nonsense in my thing. Because this here are, again, our cell names, which we have already used in order to combine, to get our tables together. We use them again here. Next, the total counts per cell as obtained from the count matrix. So let's take this thing here, layer data. Look at it. What do we get here? This is the count matrix. And if I take the column sum of this count matrix, like this, what do I get? I get for each cell the total number of reads. So let's call this total. Now our table has 2 columns. Yeah. In case I would later, in principle, it doesn't make a difference, but it has an advantage to make sure that we pull all the data out of the server object. And that is, maybe we decide to remove bad quality cells. Maybe at some point we decide cells with less than 500 counts are no good. We want to remove them from the analysis. When we would put a command at the very beginning of our script, remove all cells from the server object which don't have these. And then they would be gone from the server object and wouldn't appear here. But they would, of course, still be in the initial count matrix object that we loaded from the data. But if we make the rule that we pull all our data always out of a server object, we can always be sure that everything fits together, because the server object, if we tell the server object, if we tell server delete the following cells, it will delete it from the row names, from the cell names, and from the PCA matrix, and from the UMAP matrix, and from the count matrix. So it will automatically make sure that all these different matrices always have corresponding rows. So it's good practice, or defensive programming, one says, to always pull it from the same thing. The next thing is now the counts for one specific gene. So again, I take these here. Let's remind us how this looks like. A matrix where the genes are the rows. So let's take one gene. Let's take SLC, what had I said? 1A3. And C1A3 equals. Now I have this gene's expression. Maybe I call it simply K. That will be useful later in case we want to replace it with another gene. We don't have to change everything. And the cell pseudotime. How do we get the cell pseudotime? This is now again a little programming thing. How would you add the pseudotime to, here's a comma missing, because you see, if I want to subset a matrix, I always have to give two elements, the row and the column. And if I want to have all column, I simply leave the space behind the comma empty, but the comma has to be there. So now it works. And now I want to add to this thing the pseudotime. How do you do that? Maybe I let you think for a moment about that. Because remember, these are all 18,000 cells, but we have pseudotime only for 14,000 of these. Mm-hmm. Yes, we could do this. Then how do we subset this thing to only the 14,000? Mm-hmm. That would be, yes, we could do that. So I could take this here and say filter, that's the tidyverse thing to use filter things, and I use now something like soy dollar in Seurat clusters percent in percent lineage clusters. Problem is, now I feel, so this would be one way. I feel slightly uneasy about it because now, but let's first see, how do I now add my lambda stuff? Yeah.
Use the command on the patients of the variables, the cell total and afterwards, and then the lambdas from the PRC separate.
So just to remind us, here is this is how the PRC lambdas look like. Where would you do it? I haven't quite followed now.
I don't know, maybe I did it wrong because it didn't plot for me, but I just used like I just took the
So what I now do is, instead of a filter, I remove it, I do the same as we did before with the left join. I say again left join, and I now tell left join I want to join in this thing. That again has the advantage that it will work. But left join wants a data frame, not a vector, so I have to turn this into a data frame by n-framing it, like this. Because if I n-frame it, it's turned into a data frame. The only issue that we have here is name and value, but what I want is cell and lambda. And if I put all this together, now I have my thing as this is essentially the same construction that we used before when we tried to plot this thing here. I again used my left join to do it like that. I've written it slightly differently. You see here I have put a left join, and when I put the first table, then the second table, and then my by object. Here I have a left join, and the first table is before the left join, and the second table comes here. And the by I didn't even need, because it automatically noticed there's only one column which appears in both things. And the reason that these two things are equivalent is again this rule that putting a thing with this percent greater percent, this pipe sign before a function call, is the same as putting it here. Questions about this? Yeah.
What happens in the pseudotime rows, in the pseudotime rows for the columns that were included before, because there's just 14,000 pseudotime.
Yep, have a look. What do you see? No, what do they get filled with? Have a look. An A. This is a very important feature of the R language. It has to be filled with something. And usually, many people in many classical languages, people then put something in like a 0 or a -1 to indicate that it's missing. But this is very dangerous, because afterwards, when you have the idea of taking some summary statistics, say a mean of a whole column, then of course the mean function won't know that 0 means missing and will just count it as 0. Hence, this is an unending source of bugs and has messed up probably hundreds of research projects, that people with question, how do you mark a data item in a table as invalid? The reason why this is so difficult is because we have a specific way how we store numbers in the computer, namely 10 bytes in your computer describe a floating point number. Let's, for example, according to the IEEE 754 floating point standard, which tells you that, where is it? Here, that of these 10 bytes, the very first bit is the sign, then comes 8 bits, which are the exponent, and then comes 23 bits, which are the fractions, and they are to be read as this here is a binary expansion of a number with a, not a decimal point, but now a binary point here, times 10 to the 2 to the x exponent, and then the sign in front. Now, of course, every possible arrangement of bit in this thing is a valid number. So how can we do a non-valid number? Now, there's two solutions. Here, actually, the solution is already baked into the thing, because it's not true that everything is a valid number. The IEEE 754 standards defines a specific bit arrangement to declare certain special cases, namely positive infinity, negative infinity, and the special value NaN for not a number, which you get whenever you try to calculate divide 0 by 0 or something like that. But for other kinds like Integers, they don't have this convention that there's a specific bit pattern. For integers, every single bit pattern, it corresponds to a valid number, and hence there is no way of marking something as invalid. R has solved the issue by defining one specific bit pattern as meaning missing value, and all R's internal function will first screen for this pattern before doing any calculations. And this gives R with specific features that in all R data types, there's always the possibility to mark something as NA for not available. And this NA has an interesting concept. Let's imagine we have something like this vector here, 4, 5. And I add to this other vector, 1, 2, 3, 4. When you see, it can do this calculation, but here it says an unknown number plus 2 is still an unknown number, so it stays in NA. If I take this number here and say, calculate the mean of all, the sum of these, then it will say the sum of 4 numbers of which one is unknown is of course unknown. I can tell sum to specifically remove the NAs, and then it will give me a result. But it will do this only when specifically instructed to skip over the NAs. Otherwise, an NA is contagious, as they say. Any calculation which contains an NA will give a result NA if the NA has an influence on the calculation. So, and this is what we use here now for our lambda. This is why I can work this way of having of using this table, and all the lambdas which are unknown are marked as an NA. And we've used this already here before when we made this plot, where we gave the plot this stuff here, where the lambda was marked with the NAs, and then I gave it to ggplot and told ggplot use the lambda column to choose the color. And what does ggplot do? It decides on a color scale from black to blue and marks all NA things with a special color which is specifically selected to not appear in here. So in that way, the NA handling has worked nicely here. And this is an important point, this NA handling, which you use here. So, but where were we? We have now made here in the end our data frame, which, as you see, has now 4 columns, the cell ID, the total number of reads of the cell, the number for the gene of interest, and the pseudotime.
Is there a reason why we keep all of them?
At the moment, I just kept it for symmetry with the code before, and in the code before, I kept it because I want to see where the gray stuff is, such that I'm oriented. Here now, actually, I don't think that this is helpful, so I put a filter in the end and say, only keep rows where the column lambda is not NA. The exclamation mark is the Boolean not operator. It negates. And is NA, of course, is a function which tells us whether it is NA. That's, by the way, an interesting little mathematical issue. Why can't I write lambda equals NA? For example, if you take here these ones here, and I want to know which of these are greater than 3.5, when it says the first value is not, the second is unknown, the third one is true, and the fourth one is true. I want to know which value is 4, when it tells us only the third value is true, and for the second we don't know. What happens if I do this? Is this expected or not? Does this make sense? Because we see only the second value is NA. Why doesn't it say as false, true, false, false? Exactly. It's the whole internal logic of an NA that we can't compare it. Here we have used it by saying which ones are 4, and here it says correctly this answer cannot be set. So if I ask which of them are this unknown number, then it also does this. One says NA is in the language design parlance, one calls this an uncomparable value, which is equal to nothing, not even to itself, which is, of course, if you consider the equal sign as a equivalence relationship, you see this is an interesting exception from the general rule, the symmetry axiom, that you always have for equality, but it makes sense here. And this is why there's a specific function called is.na, which can detect the NA. Because the normal operations are designed to never say, to never give this, to never give a result when they see an NA. So this is now my data frame. Let's call this simply df for data frame because I'm now a bit lazy. And what I do here in the end, I put this df at the end so that I can get a result here. And the one thing I add here, because maybe I will need this, is this y, which is, what did we say here? Later we use this one here, log k over s plus 10 to the minus 4. K over total plus 1e minus 4. You see, I already add here this column because maybe we need this later. Okay, lambda, yes, this thing missing. So, now, what were we now supposed to do? We want to perform restrict your data frame to only the lineage cells, okay? That's what we've just done with our filter. Now perform log fit. And to do this log fit, we can now do different ways. The easiest thing is we say log fit y tilde lambda, comma, no. Comma, data frame. Could not find function log fit. Log fit is in the log fit package. Library log fit. So, now I go to this here. Could not function BS. That's in the splines package. Of course, I also know I need that one. So, and now I have one log fit here. How does this work? Remember, let's first to see what's happening here. Let's first remember what BS lambda actually does. As you can see, df dollar lambda, I have to say. You see what this thing does is it gives us here 3 different values because it makes a spline basis with 4 basis function. One falling, it makes these 4 basis function. Vlump, vlump second, third one, and the fourth one looks like this. I think we've plotted them. 4 basis function sounds a bit small because this can only give us very coarsely shaped stuff. Let's make more basis functions. So I tell it degrees of freedoms is, let's say, 8. And now we get 8 basis functions. ds, df is 8. So this was stupid of me. Now df has 2 meanings. Here it's the data frame, and here it's the degrees of freedom. And with this, now it calculates here, it says not converged, not converged, not, oh, this doesn't go good. Maybe these were too many. We go down to 6 degrees of freedom. No, it crashed even. So who has done that? who has managed to set up a logfit call at this point? My computer crashed. Session. Restart R. Error saving. This is not good. I don't like this at all. And it says error saving. Okay, now here I have this. Now it has saved it. Okay. I restart the thing now. Unfortunately, it has to run through UMAP again. But we can look here at our logfit. If we don't give logfit any special data, it will run simply a normal local regression with what have I done here? That's nonsense. Logfit already automatically does spline regression. That's why it didn't work. I don't need to put the splines. I wanted to do logfit, not spline regression. Okay. So this is what I should have done. Simply telling it my response is y, lambda is what's the x-axis, and it's supposed to do the kind of local regression that we talked about yesterday. So this is what logfit does by default. If we don't give it anything else, I think it does exactly what we did last time, fitting local quadratic splines around this thing here. So here this, now give me a moment to run through that. And with that, there's of course almost so many little tidbits about R, like I completely forgot that that you probably don't know about table joining or about NA. Of what? I think no. We have to. It should only give us an object which we can then plot. So where's this thing now sitting? UMAP. It's still working on the UMAP thing. Should be finished any moment. Is it doing our... Oh, now it's finished with everything. So let's see if this thing now gives me, yes, now it gives me a good result. Actually, if I give this simply to the plot function, yes, then it already shows me this here. We see how lambda, at the beginning, the gene is highly expressed, and when it goes down. It's mirrored, because if you look at it here, the low lambda was here, and the high lambda values were here. But as far as I remember correctly, in reality, the cells start here as stem cells and mature in this direction. Of course, our principal curve doesn't have a direction, and accidentally it got the wrong direction. So this is why this thing is now the wrong way around. But overall, this is now the result we got from this log fit. And now we wanted to do a spline fit. So this would be, so let's make a spline fit. And I first make the spline fit the easy way. I say I want a local regression of y to a B-spline basis of lambda with 10 degrees of freedom using the data frame. And now I get this here. And again, let's see if simply running plot on this thing works. Yeah. QQ. Okay, it produced us a lot of strange stuff. So I simply do this fit here, and now I can check it and see what is in this fit here. And what I have, of course, is here the coefficients, and here the fitted values. So for a simplified, simply do now the following plot df dollar lambda, comma, fit dollar fitted values. What this does is it takes for each cell, it makes now a point for each cell. This here is the cell's actual value. This is the cell's pseudo time. And this is the fitted value. So you remember, in a least square fit, you always have a fitted value, a y hat, that you compare with the actual value to get this least square. And that's what's here called the fitted value. And I simply plot that. I maybe make the size a bit smaller, and I also add points df dollar lambda, let's say y tilde lambda, comma df size is 0.2, comma cal is gray. As you see, I've switched now back from ggplot to the original plotting thing. That's simply because that's quicker to type. And once you've done it long enough, both sort of feel the same. And now you get here the results here. My curve is on below the gray points. I should turn this around. Put this first, and this here second, and points, and give this a nice color. Size, comma 1. So now here, this is the plot that we now get. Size was wrong. Compares. This is called sex. Size is ggplot. So now here, you see here our curve, which I got with the B-spline. And as before, we have this issue with this 10 to the minus 4. You remember we calculated the y with this formula here. Where was it? K over S plus 10 to the minus 4. The 10 to the minus 4 to avoid that the zeros go to minus infinity. And this, of course, means that the leverage that this thing has on the split depends on how big this value is. The smaller this value is, the further the y's get pulled towards minus infinity, and the more leverage they have to pull the red curve down. And that's why we then last time said we can use a Poisson GLM, spline fit with Poisson GLM, by saying instead of a normal thing, I use the GLM with the family is Poisson. That's also what we've done in our negative values are not allowed. Yes, and for the Poisson, I now have to put this here and say offsets like log total. Object total not found. df dollar total. So this is now the same thing, offset. And I have an offset. This is now the same thing this time with the GLM, only my fit hasn't properly worked to produce anything. So what are the fitted values here? They are fitted value on that scale. Now we come into these intricacies of R. If you have a GLM then we quickly look this up. Fitted value.glm. Now, values.glm. Extract fitted values here. Object, GLM, here it is. Fit dollar model data. Here are the fitted values, but where are these ones? Already ones. Predict GLMs. Here we have it. Link response. Should be one of response, comma, terms. Yeah.
When I tried this as well, I needed the like sample points where it was for the money. I don't know what does the term do?
Actually, I don't remember. There always used to be a. This is because it uses the offsets. So what I can do now, of course, is I can simply take the coefficients that I get from that. No, I should have prepared that. No, what's the last thing? Points. Yeah, let's do it properly. So here we have our fit. And with this fit, of course, what I can do is I can take my coefficients, which are now here, all these here, and I can multiply them with the star percent BS df dollar lambda, comma df is how many degrees of freedom did I have? 10? C bind 1 comma. So sorry that this now got a bit complicated, but x cents here. That's what I want. Steuerung C. Points df dollar lambda, comma this here, comma that now. So now I got my plot here, and this time the plot does correctly the BS. Damn it. That's not what I want. Steuerung C, Z. Sorry, I just made a very stupid mistake. Fitted values, namely, I edited here this thing. Control V. So, now we get it. Comma, ylim is C. Now, let me finish this and then I quickly explain what I've done here now. You see here now my thing goes too far down, so let's make this here a bit larger. And now here we get the curve which really shows us how SSCA1 goes down, a bit up again and down again. How have I done this now? I fitted my GLM with a Poisson loss, and I gave it here this B-spline basis with 10 degrees of freedom, and with this I can, and the offsets, you might remember from the lecture, we have to give it the totals as offsets so that they get, that the result of the prediction gets multiplied by this in order to get the case, so that the actual prediction is the percentage of all the totals which fit onto this. And now I need to get what is the prediction of this GLM for each cell. And in order to do this, let me write it slightly different when it's easier to understand. Intercept is false. No. 0 plus, and because when I don't need this here and I don't have to explain it. Intercept is false. Does it still work? Yes. Oop, but it looks different. With coefficients 0 plus does look different a bit, right? I undo it again and explain what I had here because with that it worked. So here you get this thing, BS lambda, what that one does, it takes all your, it makes a B-spline basis with 10 degrees of freedom. And these 10 degrees of freedom are now done the way, so you have 10 basis functions. And what R now has a strange idea. It usually omits the first basis function and replaces it by a column of ones. This doesn't make any difference because this is still a complete basis, only it's no longer an orthonormal basis. But for the least square fit, you still get the same result. But here, if I then want to calculate the fit results, what I have to take is here the coefficients that I get, which are the coefficients for the 9 basis functions except for the first, and for this intercept, which is the column of ones. So I take again my basis function evaluated at the lambda positions, add the column of ones that the LM function always automatically puts at the beginning, and multiply it by these coefficients to get now a fitted value for each cell. So we now have for each cell in gray the value that we got from the data, and in red the fitted value that the spline predicts. And as you can see, the red line dips under the gray horizontal line. Who does understand why that is? Who has an idea of what's happening here? I have to show you, unfortunately the screen is so small, so I have to show you the code again. So you see what's happening here is that the gray points I plotted with this Y thing. This Y thing, to remind you, is log. Maybe I just write it out. This here is log k over s plus 1e minus 4. And k over total is what I called them. And you see, this means that any cell where we have 0 counts will be mapped to the logarithm of 10 to the minus 4, which happens to be 10 to the minus 7. Would have been nice if it were minus 4, but for that I would have to use a logarithm to basis 10 here, and then it wouldn't go well with a GLM, which uses a natural logarithm. That's why I put the natural logarithm here too. And so the 0 is marked by this, but this is arbitrary. I could as easily write here 10 to the minus 6, and then the 0 line is here, and the red line does not dip anymore. So essentially, this offset here dictates where in our plot we put we indicate with 0 counts, because this is a logarithmic scale. We cannot indicate the correct position would be at minus infinity, which is not good. We cannot plot that. So we here indicate in an arbitrary way where we want to do it. And before, when we used the normal least square fitting here, we had the issue that the fit actually is influenced by this line. And if I now change in this one, the 10 to the minus 4 to 10 to the minus 6, the red line will dip down deeper. Here, however, in the GLM, in the generalized linear model, I can give it directly the counts, and the GLM won't have any problem dealing with a 0 here. Because what it does, it tries to find a predicted value, the red one, and from that predicted value, it asks what is the likelihood to get 0 counts in case the cell has 0. And of course, the likelihood to get 0 counts, even if the Poisson rate is not 0, is still a finite likelihood. If you have a process which on average gives you 1 count, it sometimes gives you 2 counts and sometimes 0 counts. So the Poisson distribution can deal with that. And hence, this red line is now the correct average over this stuff, very much in contrast to what we have here. Which shows you how important it is to use for count data the right likelihood. And maybe from a math point of view, this looks obvious, but in practice, most practical biologists, when we do this kind of analysis, they will end up with this curve. Why is that? Because what I call pipeline thinking. You say your analysis has a couple of steps. Step 1, load the data. Step 2, filter it, kick out all bad cells. Step 3, normalize it. Divide each count by the total number of counts for this cell, k over s or k over total. Next step, take the logarithm, and before add your 10 to the minus 4 as pseudo count. And then you have now these values, which I called y, which is our expression values. And from now on, work with this y. And that works well, because the y is a useful way to plot things. If I want to plot the values for the individual cells, you see I have used here the y to make these gray points. But there is certain steps where it is good to go back from the y to the original k. But if you think about your analysis as a linear framework, where you do one step after the other, and whenever you have done an analysis step, you can forget the data from the previous step because it's now used. When you do a bad job, because you say, okay, y is better when let's better work with the log normalized data column y than with the raw counts k, because the raw counts k still need to be divided. But you lose information that way. And in that case, if we do this thing, the whole thing looks much better. So now, of course, I didn't get it all to a penalized regression, but also we didn't do anything new in the lecture yesterday. So with everything that I've shown here now, I will upload what I've done here afterwards, when you can work through it and when you can try the penalized regression again. The one thing I wanted to show you is the heat map. We can, of course, you mind if I still spend 5, or maybe you can try, if we can think about the heat map, we can think manually for a moment. So remember, in the lecture, we used, where have I put that? In the end, we made this heat map here. And now we want to do this heat map again. We want to reconstruct it. How would you do this now? One among all these things is somewhere the gene that we have now looked at, SSC1A3. How do we now get this heat map? What would you say is the first step? What kind of data object do we need to make this heat map? What data is below that? Below that is a matrix which calculates a matrix where each column corresponds to a lambda value and each row is a gene. And what we've currently calculated, which should be one row in this matrix, the SLC1A3 gene. We have done it a little bit lazily, because what I've done here is because I simply use the values, the lambda values for the genes. as we from the cells. But strictly speaking, what I should do to get a nice red line here, you see here something missing in the red line, because these points, it jumps here, there's no cells, but I still would like the line to go through here. So what I should have done is, instead of inserting again the lambda values of the cells into my spline thing, I should have made again a linear grid of values which scale from 0 to 200 in steps of maybe 0.1, and calculate for each of these, or in steps of 1, and calculate for each lambda values 0, 1, 2, 3, 4. I put this in and I get a corresponding y value. So in that case, if I say for each of the lambda values from 0 to 200, for each integer value I calculate one red y value, then in that case I get 200 y values, or 201 if you do that. And I can do this for a whole list of genes. And this whole list of genes I can then arrange in one big matrix. And this matrix is matrix then, no, and then this matrix I can give it to a heat map function. It will plot this heat map. Now here, if you then try this maybe again for next week, and then we can talk about this, there's a little practical problem when we now do it in R and you're not so used with R, so how to deal with that. And there's two possibilities. One possibility is you switch over to Python, because all this can be done in Python as well as in R. And if you prefer that, you can do this. Another idea is that you say, I now know conceptually what I want to do, but I don't know the exact code. So for example, to make the heat map that we've just had, you might say, okay, it's easy for me to, I can, for example, say, let's write a function which takes a gene name and then produces this vector of 200 y values. And to do this should now be possible by just taking the code that we have done here and putting it all together in one big function body. So then you have now a function which takes a gene name and produces a vector. And you also have a list of genes which you want. You can simply take the 20 highest variable genes or so. And now what you now want to do is call the function for each of these 20 most variable genes and arrange the results in a matrix. And once you have dialed it down to this specific task, it's rather easy to use something like ChatGPT. In that case, what you would do, if you are not that familiar in R, because after all it's about the concepts and not the details of one language, you could now tell R something like, I have a function which takes a gene name and produces me a vector of fixed length. I want to call this function for a list of different genes and arrange the result in a matrix. Give me the code, and then it will do that. And you just have to replace it will write as apply your function or something like this. Then the next time you might come into the problem that you say, okay, now I need a list of gene names, and the problem sheet said, use the 20 most variable genes. And how do you get that? If you remember the server thing at the very beginning somewhere, it actually finds the most variable genes here in that step here. You can now try to find out how to get it out, or I can also go to ChatGPT, and if I now ask, how do I find the most variable genes from a server object, it will tell you all kinds of nonsense. It's probably better if you specifically say, I have a server object for which all the usual functions have been run, and I want a character vector with the names of the 20 most variable genes. That's something that ChatGPT should be able to figure out. It has its problems with server because ChatGPT gives you a good result for stuff which is well documented on the web. All the things that you won't find by Googling, ChatGPT won't know either, obviously. And the server documentation has a lot of gaps. But I think they filled it in recently, like in the last year. And let's hope that ChatGPT has already read the finally completed server documentation. Otherwise, it won't help you with that. But I think I tried that recently, and it now knows how to find such stuff. Okay, so I would say use this, give it a try, or do it in Python if you want to do, and see if you can sort of get this stuff and add to it the fact now to switch over from the spline regression that we've now done here with the basis splines to the specific p-splines with this tri-diagonal penalization matrix. And the one thing you will notice is that this standard GLM function that I have here doesn't take a penalization matrix. That's why I put at the end of it this explanation how you can use this GLM GP package, which does take a penalization matrix, but otherwise does the same as the built-in GLM thing. So last thing for today, I told you about the thing here. If I now click on render, what it will now do is that it runs all the code again with a freshly, newly started R and produces an HTML file with the output. There's two things which you need to know with this rendering. The first thing is it reruns your code with a freshly, newly started R where nothing is loaded. And that actually can have some unexpected consequences, because in your practical work, what you keep doing all the time is you change something in this chunk, when you go a few steps up, change something in that chunk, when you decide to execute something here, when this chunk, when the chunk up there, and in the end you haven't executed the chunks in the order in which they are listed, and hence haven't noticed that actually the chunks don't fill the variable in the proper order, and the chunk that uses variable x is farther up than the chunk that fills it. So whenever you work for an hour on such a notebook and then try to run render, what will always happen is that it tells you I can't find variable x, and this is usually always because you have arranged this code wrongly. The best way to debug this is to put your R into the starting thing by clicking here on this broom symbol to delete all variables, and then going here onto the restart art to also kick out all loaded packages, and then click on the little green arrow of all your chunks in order. Execute one chunk after the other, and when you execute them in the order that the render function will later execute them, you will be able to debug this. Then, finally you have a nice HTML file that you can send me as an email, and I'll be happy. But this HTML file will miss all the images, because what it actually will produce is the HTML file and a subfolder in which it puts all the images, and the HTML file will only contain references to that. And you would have to send me the file together with this folder, but I don't want to get so many files. So what I would ask you to do instead is to look, if I put that, to look here, where you see that at the very beginning of your of your Quarto notebook, R has put in this, what it calls a YAML block, in these double things. In this YAML block, you extend with, where was it? You extend with this thing here, written exactly as it says here, with exactly this indenting. And then this tells R to embed all resources, so put all the images into the HTML file and really produce a single file which contains everything. Yeah, you can click on it here. So let's give this a try and just see how far you get with this, with getting this all. I realize this is all a bit, actually it is difficult, because if you do it in R, when you have to get used to a new language which has a very strange interface. If you do it in Python, you have to sort of pull over all the stuff from the code into this part. Maybe I should have made the lecture notes in Python, but then I have to restart with everything. So let's see how far you get. And if you run into any questions, we could also consider making up a Moodle forum where we can exchange, where you can ask questions, people answer it. Any interest in that? Or do you have something better than Moodle anyway? I'm not quite sure I can see if I can ask them to give us a Moodle thing, but I've forgotten who to ask for that. These Moodle sections always appear out of nowhere. You suddenly have a Moodle thing for your course and you don't know who put it where and why. We'll see. I can try to find a Moodle, but when you all have to enroll and we have to do an enroll key, and I find this all a bit cumbersome. So just send me an email if you have a question. Okay, so that was about this. I hope you've now got a bit of feeling how to work with R and with tidyverse and how to work with tables, which was not at all what the homework was about, but which is, again, of great practical relevance if you work with real data. So try it out. And as I said, I will now upload this thing that we've just developed together today and add it to the webpage. Other questions, remarks, other stuff? Then see you. Oh, right. Next week, next Monday is Pentecost, I think. In case I'll be in Tuesday, so when maybe this thing that we've just done with doing new stuff we can discuss on Tuesday, which would even have the advantage if we make an exercise on Tuesday and no lecture on Monday, that we finally get in sync again, and because currently the exercises are always 1 week behind the lectures. So let's do that. So see you all on Tuesday next week here.